I'm going to introduce our plenary speaker, our keynote speaker, Jennifer Schneider from Centauri Therapeutics. Now, this company is really interested in developing innovative solutions and anti-infection therapies. So it's a great pleasure to introduce Jennifer. Thank you very much.
To improve outcomes with those with difficult to treat infections. And I'll repeat this theme over and over, but when we think about infection, it isn't just antimicrobial resistance. It's all the people who have theoretically treatable infections, but because of any variety of comorbidities or because of cancer or because elderly, that they're not responding well to treatment. So when we think about the types of infections that need to be treated, they are those classically resistant infections, but it's also this much broader need that exists. So I wanted to start by talking about the team. So, doing the work with a creative and resilient team. And I wanted to start by talking about the team because I remember about 25 years ago when I got started working, I was hearing a venture capitalist from Kleiner Perkins talk, and he said, before they even look at the technology, they look at the team. And I thought, well, that can't be right. You must think about IP first. You must think about, what the exit is going to be. And, I just didn't think that could be true. And now, having been in this industry as long as I have and seen companies succeed and some companies fail, it is the team. And so I'm really, really proud of the team that we have at Centauri and proud of their resilience, proud of their creativity. We're currently about 16 people and growing, which is a really nice thing to say. There are 7 lab scientists up at Alderley Park, so pleased that we have so many people who are able to do that fundamental lab research. We have other folks around the UK, Switzerland, and the US, and we have a strong team of consultants as well. So if you think about building a company or building a lab, don't forget all those different resources that you can build on to ensure that you can do that work that you want to do. And our team, I think, has been successful because we've got people like me who are strongly rooted in biotech, so I'm one of those people who has to wear a lot of hats on any given day, and I'm used to fighting fires, and that's my job. But our team also has a lot of people who've come from big pharma, and I know that sometimes we think about pharma as big business, lots of silos, but this team that we've been able to build who do have this pharma experience, I often think about our CSO, our CMO, our chairperson is kind of like Mary Poppins. They're able to pull all these tricks out of a bag when I need help, right? I'll be facing some issue and they'll have some skill that they learned while they were being trained in these organizations. So really appreciate the types of learnings that they're able to bring to the table, particularly in the biotech sector where our resources are thin. We need people to be able to come to the table and bring all of these tools to help us all succeed. We've been very, very fortunate in terms of our resourcing. So we have raised a significant Series A, so 30 million pounds to date. But most importantly it's the company on the bottom left, Animatrix. So this is a family office, a group of high net worth individuals who, when there was just this idea, and I'll tell you about what the work is as well, but when there was just an idea, this family office came to the table and put money towards this company so that we could get going. And we also received £1 million from UKRI. And this was at a time when we were really just trying to get some data so that we could go out and fundraise, so that we could put slides together. And so as I said, I'm not big pharma trained and I don't have many good management stories, but one of the ones that I think about a lot is years ago I was watching a program and they were talking about how they built the Dreamliner, the 787. And this whole episode was about the landing gear on the 787. And if they just reduced the weight on this landing gear by this fraction of a percent, that they could reduce the overall weight of the plane by, this many pounds, and which meant that they could carry a little less gas, and they could carry a few more patients, and they could go a little bit further. And I think about that with our company all the time, that, first of all, that every job is important, every person is important, everything that people bring to the table every day is important. It's easy to think, well, I'm just this little cog on this little wheel on this airplane. It doesn't actually have an impact, but actually everything has an impact. Just like the original money that we received from Animatrix, significantly less than 30 million pounds, but it's the reason that we're here today, and it's the reason that our company is on the verge of being in the clinic. So don't discount those initial few dollars that might enable the research or enable the work that you're looking to do. And doing the work through tenacity, I think it's just really, I think everyone here knows how hard it is some days to show up and keep labeling tubes or keep pipetting or keep doing this thing over and over. And we all do it because we've got, I imagine that we all have like some underlying reason to want to do it. This is really hard work. And I don't think we do it because we think we're going to get rich or because, there wasn't anything else we could come with. We really care about this work. And for me, I grew up in Singapore, and I had a health teacher who I thought was so cool. She had come to Singapore from Geneva. She'd worked for the World Health Organization. And so I decided at 10 years old that I was going to work in public health, and I was going to marry a Swiss ski instructor. That was the plan. So ergo the picture of the Swiss train, but that was the plan at 10 years old. But one of the other things that I learned in Singapore was, that was the first time, my family is from the Midwest, the United States, so farming background, very, very white bread, very all-American family, and we ended up in Singapore. And it was the first time that I saw health disparity. I'd never really seen a yellow vaccination card before, and I started to get vaccinated for things that I'd never heard about in the U.S., and started to see disease in a way that I hadn't seen it in the U.S., and it led to a question in my mind about, well, why do we invest in some things and not other things? Which led me to study dengue for my PhD. So the picture from the West Indian Tobacco Company is from Trinidad. So I did my PhD studying the dengue viruses in Trinidad, which then led me to 20 years in Washington, D.C., where I worked with companies to try and find funding, to develop partnerships, develop collaborations, to enable work in areas of unmet medical needs. So funding for things like malaria, dengue, pandemic preparedness, biodefense. But it also led me to anti-infectives and antibiotics, because a lot of the market challenges with antibacterial development mimic some of the same sorts of market challenges with other global health needs. And it was through this time in DC that I heard a story that led me to Centauri Therapeutics. So I will tell you the story that I heard. I'm like, this is a really cool technology, and I want to be a part of it. So this is the work that we're doing at Centauri. So I mentioned Animatrix founders, and the guy at the top right is Rupert Lywood. He's the individual who originally bankrolled our little company. Stu still sits on our board, who works for Rupert, and then Mike was our original CEO. And the three of them found a technology that Carrie Mullis, who was the inventor of PCR, had. So Carrie had this idea. We all have these, we have this massive antibody repertoire. Why aren't we able to put some pre-existing antibodies that we all have to work? Why are we just letting them sit around? And that's the origin of our company. The antibodies that we're looking to take advantage of were first identified in the 1960s when physicians tried to use pig tissues for heart valve transplants. And while they were prepared to manage a graft versus host type response, they instead found an almost immediate rejection of this foreign tissue. And they realized that the reason this tissue was killed off is because it was coated with, in this case, alpha-gal sugars. And the technology then is all based on mimicking this sort of xenotransplant context. Can we coat, whether it's bacteria or a fungus or an oncology cell, can we coat that thing with enough sugars that we're recruiting these antibodies that were, like I said, identified originally in the context of xenografts. And when I heard this story as a consultant in DC, my first thought was, this is a lot different than a beta-lactam, beta-lactamase type product. And my philosophy has always been, this is the kind of technology that deserves the chance to fail. What I mean by that is, to get a drug into the clinic, I mean, we're on the verge of having spent, I don't know, $50 million in total so far, and we're on, to get to the place we are, right? But you need all of that money in order to advance a program, in order to have other molecules fail. But I just thought, if we're really going to do anything different in this space, if we're really going to try and get out of this cycle of resistance or address infections in a different way, it's going to take a lot of money, it's going to take a lot of creativity. We really have to put the pedal to the metal and see what this mechanism might be able to do. Can we kill off bacteria as rapidly as you were able to kill off a xenograft? So that's the work that we're doing, and that's the origins of the platform. Importantly, these sugars these antibodies that we're looking to take advantage of, they're ubiquitous, and importantly, they're really, really hard to get rid of. So we know that even in severely immune compromised individuals, these antibodies are still there. So I'm not going to go through all the wonderful things about these antibodies, but when we think about addressing the issue of antimicrobial resistance and undertreated infections, the fact that these antibodies should be present, including in these difficult to treat patients, gives us a lot of reasons to believe that we have the potential to have efficacy in that context. So I'm sure this is all data that we all know, but this is why we really care about those types of patients in particular. So I know we're going to talk a lot about antimicrobial resistance today, but it's also important to remember that, for example, in the recent phase 3 studies for UTI and HAP-VAP, even in patients who were screened for sensitivity and resistance to both the standard of care as well as the intervention drug, there was between 35 and 50% clinical failure in those contexts. So we know that a good portion of those people did not respond, not because of the sensitivity of their bug, but due to host factors. And we see this over and over and over again. We know it's true with cancer patients. We know it's true with people who've had transplants. We know it's true for people who are on antibody therapies. And so, I just think when we talk about antimicrobial resistance and think it's this little thing, it's actually a huge thing that is impacting all of us all the time, our friends and neighbors. Anyway, we think about antimicrobial resistance in terms of that sensitivity resistance thing is just the tip of the iceberg. And there's a lot more going on that we need to think about treating. We know that traditional approaches to infections are no longer sufficient for many patients, and that people are now saying this out loud more and more and more. This is Dr. Steve Holland from the NIH, who was speaking at ACMID in 2024, and he said, we've all been there. We have the bug, we have the sensitivities, we're giving the right antibiotics, we even know the patient is taking them, and they are not getting better. And this is where host factors come in. And the NIH recently published an article in Nature saying the same thing, right? That 94% 94% of deaths and over 99% of serious infections are associated with organisms that are susceptible in vitro to currently available therapies, but patients are not responding, and this, again, is because of host factors. So we, and I think others who are trying to do non-traditional things in the antibiotic space, we all have the opportunity to transform anti-infectives in the same way that immune oncology has transformed the way cancer care has changed. So, and we think just like with cancer care, right, chemotherapy and radiation still do the heavy lifting in that context, right? But immune therapy has really changed the outcome for a lot of patients. And we think the same thing for the antibacterial space. We know that the beta-lactams are going to do the heavy lifting for a long, long time. But we also know that for a set of patients, having a different modality is going to impact outcome. So one of the things that I talk to people a lot about who may not know this space well, in patients who have comorbidities, the antibiotic is actually doing exactly what it was designed to do. The antibiotic comes in, the bacterial load drops. The problem with these patients, though, is the immune system isn't able to come in and do the rest of the job, and that's something that we count on when we build antimicrobials, whether antivirals, antibiotics, that the immune system is going to do part of the work. And when that can't happen, even when the drug is totally doing its job, you're left with these recalcitrant infections, these chronic infections that just don't go away. And so we're hoping that by bringing the immune system to bear, by taking advantage of those pre-existing antibodies, that we can change the outcome for these patients. So I cared about this technology, this company, for a long time. Over the pandemic, I helped the company raise our Series A, close the Series A in 2022, and this is our original team. And since I know that a lot of you are bench scientists, this is something that I think a lot about and talk with our team about. But David Baltimore, who won the Nobel Prize and died last year, he made a comment. He said, when you are a scientist and you're trying to prove or disprove a notion, you work at the bench doing the dullest, most routine things over and over and over again. I can't tell you how many ways things go wrong all the time you are doing this because there's an idea behind it. And that's how I feel about our work, that we all care about this mechanism, we all care about the patients that we're trying to address, and you have to show up day after day and put your heart into it, even though you think, God, how are we going to get through this challenge? How are we going to overcome this next thing? And working with a non-traditional agent, we've had to be really creative. We've had to come up with new ways to do things. And I'm going to show you two pieces of data because this is a science conference, so this is one of them. The reason this slide is important is this actually represents 3 years of work. What you don't see on this slide are all the animal models we tried that didn't work, all of the different in vitro assays we tried that didn't work. We really wanted the things that everyone else used in this space, like the neutropenic mouse model. We really wanted to put that thing to work and be able to show off how our molecules sing and dance, but it didn't work. And so, the number of studies that we brought to the table, like, God, what did we do wrong this time? Did we not get the titer of this thing right? Did we not dose the pathogen right? And finally, when we got this data point where we were able to demonstrate the added value of the antibodies that we're looking to take advantage of and how that improves survival, to us, this was a massive win. The other thing that was a huge win for us, this was our light bulb moment. This was my attempt to be creative. The other thing that really was a huge shift for us was actually moving away from animal models altogether and into a whole blood assay. And because it's in this assay where we're actually able to demonstrate the totality of the effect of the molecule. And more importantly, again, than the data, it's this team that comes to work every day, that has to fight through the highs and lows of science. We're able to find this assay and develop it, put it to work, so that we could show off the cool things that our molecules can do. And so I'll just tell you, let's see, perfect. So just in a bit of data, for those of you who are familiar with MIC, this is our molecule if you were to do a traditional MIC assay. So this is just in our PMI buffer, and you can see what the potency is with that. If you add our molecules into an assay where you include human sera, we can also look at the added benefit of complement. So you can see the increased potency when you bring complement into the system. But when you put the molecules in whole blood, so where you've got neutrophils and complement and phagocytes and the antibodies that we're looking to take advantage of, you can now capture the totality of the effect of the molecule. And this was a huge win for us, a huge shift in not just our thinking, but in the conversations that we were having with other people, including the regulators. So when we took this assay to the regulators and said, look, the mouse that you're likely familiar with does not actually show anything that we need to show you about our molecule, but this assay does, they said, we agree. And to get a we agree from the FDA, I felt like I'd won. You know, that was just the most amazing thing. But back to the point about doing the hard work, it took, again, years and years to get us to this point where we have data to show, data to take to the regulators. So just, I think, as any of us are, you know, going through our days doing the hard work, having that tenacity, having that creativity is just critical to going forward. And so just finally, you know, leads me to our purpose at Century. You know, this is the purpose. This is this is a slide that we always have in our slide decks. You know, we seek to tackle life-threatening infectious diseases in the most vulnerable patients, and that's the work that we're doing. So thank you.